A powerful natural substance, once extracted from corpses to treat patients, is now being engineered in cutting-edge labs. This substance that is so powerful it fuels growth, strength, and recovery is now mass-produced on an industrial scale. Sounds a lot like something out of a sci-fi thriller, right? Well, it's not. With the soaring demand for human growth hormones, scientists have cracked the code to synthesize this life-altering compound, redefining medicine, athletics, and even aging itself. Let's first learn the origin story and how they work in your body before breaking this process down into something we can all understand. The human growth hormone is a substance secreted by the pituitary gland, which is located just below the brain. By combining different amino acids, the pituitary gland is naturally able to produce the growth hormone, which is in charge of stimulating the growth of all tissues in the body, including the bone. Scientists have known about growth hormones since the early 20th century, but it wasn't used for any treatment until 1963 when they started getting used to treat children suffering from pituitary disorders, which made them unusually short. But the drug was in short supply because its only source was from a human corpse. Collecting the substance meant isolating it from the pituitary glands of a corpse through a complex, labor-intense process. Even though this technique made it hard to collect, it continued for decades. But one day everything came to a stop, with the horrifying discovery made in the 1970s, which showed that some of the extracted substances were contaminated, having been extracted from a corpse infected with Creutzfeldt-Jakob disease. This disease caused rapid brain degeneration, leading to death within 12 months of when the first symptoms appear. Within a few years, this tragic discovery led to the death of several infected patients. The fact that there was no way to test for it turned the lives of all of those who had previously been treated into a terrifying waiting game, in constant fear of the onset of symptoms. But it wasn't until 1985, 15 years later, when the U.S. Food and Drug Administration finally stopped all distribution of corpse-derived HGH. Coincidentally, during the same period, scientists had started exploring new ways to obtain the hormone. A biotechnology company called Genentech and a drug company called Eli Lilly had both independently been working on a new way to artificially manufacture HGH using recombinant bacteria. We'll break down the how in just a minute. Soon enough, after the breakthrough, the FDA approved Genentech's recombinant HGH in the form of an injectable drug called protropin. The recombinant, meaning genetically engineered, human growth hormone, works very similar to the natural HGH produced by the pituitary glands. Since the rHGH isn't produced in the body, it is administered through subcutaneous or intramuscular injection. Once injected, the rHGH enters the bloodstream and sticks to growth hormone receptors, which are found on various cells throughout the body, especially in the liver. The liver responds to rHGH by producing insulin-like growth factor, IGF-1, a key player in HGH effects. IGF-1 then circulates through the bloodstream, driving anabolic effects like muscular growth, stronger bones, and cell regeneration. It also influences metabolic effects, such as breaking down fat, increasing insulin resistance, and regulating blood sugar levels. In short, mass-producing the human growth hormone in a laboratory is like teaching tiny living factories to make the hormone. Scientists give them special instructions by means of a DNA gene code that tell them how to build HGH. These tiny factories then grow and multiply, producing more and more of the hormone. After that, scientists clean and purify it so it's safe and ready to be injected into the patient. Once inside the body, the growth hormones do their natural thing to aid the body in cell repair, muscle growth, breaking down fat, and more. Now, let's go step by step to see how these hormones are made. Step 1. Select a suitable host system. A host system is the biological factory used to produce specific proteins. It includes a host cell and the genetic instructions needed to make the HGH protein. The host cells can be a bacteria, a yeast, or mammalian cells. Scientists choose the best system based on the efficiency, cost, and the type of protein modifications required. Scientists typically use one of two systems: the E. coli bacterial system, which is the most common method because it's efficient for large-scale production and relatively inexpensive, or the mammalian cell system. This is used when additional modifications to the hormone are needed, but it's more complex and costly. These host systems factories provide the environment where the hormone can be synthesized and developed. In other words, the cells follow genetic instructions to assemble the hormone, linking amino acids together in the right sequence, just like a factory putting together parts to build a final product. Step 2. Prepare the modified plasmid. To produce HGH in the lab, scientists first need a carrier to deliver the HGH gene into the host system. This carrier is a small circular piece of DNA called a plasmid, which works like a genetic USB drive, carrying new instructions into a cell. To insert the HGH gene into the plasmid, scientists use restriction enzymes to cut the plasmid open at a precise spot, like a pair of molecular scissors. Then, DNA ligase pastes the HGH gene into place, making sure it fits perfectly in the sequence. Think of this gene insertion process like editing a software file on a USB drive. You first delete a section of code to make space for new information, then you paste the new information into the code, then you save the file. Step 3. Insert the plasmid. Now, the newly modified plasmid is ready to be inserted into the bacterial computer to start producing HGH. Once the bacteria receives this new genetic software, they follow its instructions to produce HGH. It's kind of like a computer reading files from a USB drive. Just as a computer follows instructions to run a program, the plasmid acts as the USB drive, and the bacteria or cell system serves as the computer. Heat shock, or electroporation, are two methods scientists use to get the bacteria to accept foreign DNA. The foreign DNA being our HGH-induced plasmid in this case. Step 4. Grow HGH at scale. Once the plasmid is inside, the bacteria read the new DNA instructions and start making the human growth hormone. In this stage, the transformed bacteria are grown in large fermentation tanks under controlled conditions. They manage the temperature, pH, oxygen, and nutrient levels. Once they reach a certain stage, the bacteria starts producing human growth hormone as they continue to multiply. To enhance the production of HGH, a chemical inducer such as isopropyl thiogalactoside is added, which then signals the bacteria to produce the HGH in larger amounts. Step 5. Harvest and purify the rHGH. After a specific amount of time, anywhere from 12 hours to a few days, depending on the system, the bacteria are harvested and broken. The contents of the cell, including the rHGH, are then released into the solution. Since bacteria produce many different proteins, the rHGH must be separated and purified to ensure it's safe and effective. A good way to understand this is like making fresh squeezed orange juice, but instead of juice, we're extracting pure HGH from a mix of proteins. Scientists use a combination of methods, choosing the most effective steps based on purity needs and efficiency. Once this process is complete, the rHGH is ready for medical use. Step 6. Testing, quality control, and packaging. Once the rHGH has been successfully harvested and purified, they are put under strict testing procedures to ensure their purity and effectiveness. Without detailing the testing procedures here, it's enough to know that the molecular weight and purity of the rHGH are confirmed, as is the structural integrity of the rHGH, which help ensure rHGH functions like the natural HGH in the endotoxin test aimed at ensuring that no harmful bacteria toxins remain. Once the rHGH passes all the tests and is considered safe for use, it is formulated by mixing stabilizers and preservatives to increase shelf life. It is then freeze-dried into a powder form, and the final product is packaged into vials. Before injection, it must be reconstituted by mixing it with a liquid, which is typically sterile water or sometimes bacteriostatic water. This stack water helps to dissolve the HGH powder into a solution that can be easily drawn into a syringe for a shallow injection into the fatty tissue. Pop quiz. Do you remember the 6 steps of making HGH in a factory? 1. Select the host system. 2. Prepare the plasmid carrier. 3. Insert the carrier into the bacterial host system. 4. Let it grow inside the host system. 5. Harvest and purify the final product. 6. Test it and ship it. For more simple breakdowns like this explanation on the human growth hormone, be sure to subscribe so you stay up to date with this educational content. It's free and it helps us bring you more interesting videos. Check out our health and food playlist of other factory creations like the science behind making breast implants and how plant-based meat is made. And of course, if there's a product or trend you would like to see covered, please let us know in the comments below. Thank you for watching.